Hi, class. Okay, here we are going to do 723. It says a female Drosophila heterozygous for 3 recessive mutations, ebony, scarlet, and spineless, were test crosses in the following progeny were obtained. This is not, this kind of looks like the data set that you see on 723, but this is how I have reorganized it. I always like to, in the beginning, rewrite the data set, bringing the most numerous F2 individuals to the top of the table and the least numerous here to the bottom. Early on in the chapter, they would actually organize it that way for you. And the reason that they did that is because it's a lot easier to see what's going on. So on 723, they didn't do that for you. It's giving you more what it might look like if you were in a lab collecting that data. They also would just write like wild type, and they wouldn't specify wild type for each of these genes, right? Or the alleles for each of these genes. And so ebony is body color, scarlet is eye color, and then spineless is the bristle type, right? And so so for here, they would just say on this row, they would just say wild type, but I'd like to like actually explicitly spell it out because it's easier for me to get it, to keep it organized. And so then this top one right here, this 347, they would just label ebony spineless. So you can go to your book and see where they say ebony spineless. That's the one where you have 347. What I do is I say, okay, I'm going to write it out, all of them out. I'm sort of going to placehold every individual allele at each of those genes. And it just makes more sense to me. And so I've done that here. So the reason that I took the most numerous and put them up to the top is because the most numerous ones, if you remember those F2s, are the parentals when the genes are linked. So first off, we can see that, okay, there's a massive variation in the number of F2 individuals for each of these phenotypic classes. And the reason is because the genes are linked, right? If the genes had not been linked and we'd done a test cross between sort of a trihybrid test cross, our expectation would have been a 1-to-1 to 1 to 1 to 1 to 1 to 1 to 1 ratio at each of those phenotypic classes. If you remember the dihybrid cross, it was a 1-to-1 to 1 to 1 ratio, and the monohybrid cross test cross was a 1-to-1 ratio. We don't get that, so that already answers part A for us. We don't get that. That's telling us that something else is going on. Not only do we get a deviation from that ratio, from that test cross ratio, but we also see that the parental, right, types are in high percentage. Okay, so the parental type, the parental was a trihybrid. So if you took these two phenotypic classes and kind of merged them together and just treated them as chromosomes, you'd see that you have 3 genes, right, that are heterozygous at each of those genes. That's the parental type, right? Because there's an overrepresentation of that in the F2s, that's telling us that, okay, these genes are linked and that they aren't assorting independently. Remember too, the least frequent one represents these two down here represents the double crossing over. So there's been a crossing over between all 3 of those genes. And that's why I put them down there. So here it is reorganized like that. Okay, so our first thing, well, actually, we can even look at the question 2, what is the genotype of the original heterozygous individual? And we can see here, this is what the genotype would look like, right? Because we have these 2 different phenotypic classes, this now tells us the phase of them. So the phase in that original F1 individual, she had a mutant ebony wild type allele, spineless mutation on one of the chromosomes. On the other chromosome, she had wild type, scarlet, and then wild type. Okay, so that already tells us that. So our first order of business now, though, is to figure out which of the genes is in the middle. All right, and so how we do that, if you remember, we look at the parental, and then we look at the 2 least frequent ones, and we see which of the alleles is sort of flipped in the double crossing over, because that's what it does. What happens in a double crossing over event is it flips the middle gene, and so because of that, we can identify it. All right, so we see here we have ebony wild type SS. Here we have ebony wild type, right? But then wild type. So we see here that that SS is flipped. We can also do the same here. We have wild type, ST wild type, and then here we have wild type ST again, but then here we have SS. Again, identifying that as the flipped gene. Therefore, that tells us that the scarlet gene, right, that gene for eye color, is in between, is located in between the ebony gene and the spineless bristle gene. Okay, so now, rather than just keep going, I like to go really slow and just now rewrite it, but reorder those and put the spineless gene in the middle. So here I've gone through and I've rewritten all of those. So here, ebony wild type SS, I say ebony SS here in the middle, wild type. Wild type ST wild type, here's wild type, because that's the spineless one, and then ST, that's on the end. Okay, so here again, this is what this is. So in this cross, because it's crossing with a recessive male, the gamete that looks like this crosses with a recessive male, and you get this exact same phenotype. Same with this, the gamete that was wild type ST crosses with an ESS ST male, right? That the males are giving that to every one of the gametes. And so you get that phenotype out of it, wild type ST. So this is what happens when there was no crossing over. These are the phenotypes that result when there was no crossing over occurring in the production of the gametes in the female. These are the ones that, these are the phenotypic classes that were produced, the F2 individuals that were produced when there was a double crossing over. So there was a crossing over between, so let's just take these two phenotypic classes and just pretend these are the two different chromosomes, right? And so you have a crossing over here between E and SS, that gives you the E down here, the wild type up here. You have a crossing over between the SS and the ST, that gives the wild type down here and the ST there. This after that double crossing over one, this one then becomes this one here on the bottom, and then this one then after the double crossing over becomes that. And this is how we can see, this is where we can see that that middle gene is flipped. So that's the double crossing over. So now let's look at this. What this one shows us, what happens if we have a crossing over between here and here, right? Oops, sorry, I didn't mean to do that. Hopefully that doesn't mess it up. Cancel, never mind. Okay, so crossing over, so let's just put a crossing over here, right? That'd bring the ST up here and the wild type down here. That's what we have here. So that's showing a crossing over. These are the F2s that resulted from a crossing over between the SS gene and the ST gene. Here are the individuals that were the F2 individuals that resulted from a crossing over from between the ebony and the scarlet here. Okay, and so if that didn't seem apparent to you right there, or take some time and look at that, and even flip them around, convert these two F2 phenotypes into two different chromosomes, and then just cut them and flip them and see how all of that works. Okay, but now we're ready to answer the questions. And so here's spineless C, spineless gene, or sorry, I was looking over here. The spineless gene here is in the middle. So now this first question asks, okay, what is the distance between ebony, right? Ebony here, and ST all the way over here. So these are the two genes that flank this whole thing. And so we have to figure out, okay, how many crossing over events in our data did we observe to help us to estimate the distance between that? And so here, so this is like basically all the crossing over events that have occurred. So in these 67 individuals, there was a crossing over event between E and SS, right? These 76 individuals, there's a crossing over event occurred between E and SS in the production of the gametes. And so that's a crossing over event between E and ST, right? Because ST is over here. On these 68 individuals, this shows a crossing over event between SS and ST, but that also means that there was a crossing over event between E and ST, and the same down here. So you add 67, 78, 68, 503, And in these double crossing over events, there were actually 2 crossing over events that occurred between E and ST, right? And so you had here, add these 2 up, but then you would times it by 2 because they're double crossing over events. And so that's what I've done down here. Add all of those up, divide them by 1,000. That equals 0.303 or 30.3 centimorgans. That answers question D. Question E, between E and SS, we want to get we want to see the count the crossing over events that occur between E and SS. So here, E and SS, we see that it is these. Here's a CO2, the crossing over, the second crossing over. So here, it's E and SS, 68 of them. E and SS, the crossing over event occurred between here to produce that. That's 54 of them. And then here, E and SS, and here E and SS, we're not counting the second one this time, right? Because we're just looking at the crossing over event between this gene and that gene. So we're only counting 8 and 18. You add all of those up, 140 over 1,000 equals 14 centimeters. Okay, or centimorgans, sorry. 14 centimorgans. Okay, so that answers 2 for us. Now, F says, what is the coefficient of coincidence? Coefficient of coincidence equals the observed number of double crossing overs over the expected number. Now that we have those, so we have 10 and 18, right? Those are the distances. I mean, those are the observed numbers, right? So that is 10 plus 8 is 18 divided by 1,000. That is 0.18. Here we have to now get the distance between each of those genes, right? So here we have the total distance between the genes. Here we have the distance between E and SS. In order to get the distance between SS and T, we just subtract this one from that one. That gives us 0.16.3. So now we have all the distances between the genes, right? This was the total distance between the flanking genes. And so now we have the ones between the interior ones. And so in order to have the probability of a crossing over event occurring between E and SS is 0.14. Crossing over event, the probability of a crossing over event that are occurring between SS and ST, then, because we minus it from that, is 0.163. We add all, we complete that equation. That equals 0.789. That is a coefficient of crossing over. We can put that into our interference equation. It didn't ask us this, but we could do that. And then the interference would be 0.211. Okay, so now for E, they just want us to diagram it. We've already diagrammed the cross. We've already looked at up here at the no crossing over phenotypic classes, and that just basically tells us what the F1 heterozygous female looked like, right? That was crossed in with the male that was mutant, 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 and then the remaining crosses gave us all of these phenotypes, and we could just add the contributions of the male, the mutant, mutant, mutant to each of those, and that would give us the genotypes of all of the 8 individuals in the 8 different phenotypic classes. Okay, so that's it. That was a little bit long, but you're not going to have a more complex type of 3-point cross on the exam, more complex than this one. This is sort of the upper limit of the complexity. All right, good luck. Let me end this, and good luck studying.